Welcome to the Dirt on Growing, where we don't just plant seeds, we plant ideas that challenge the norm. I'm your host, Brandon Rust from Full Crop Sciences, and this is the podcast where living soil meets real science. From worm castings to bottled nutrients, microbial inoculants, and chemical pesticides, we're here to question the trends, break down the data, and empower your grow. It's time to think deeper and grow better.
What's going on, everybody? Brandon Rust here. Welcome to another episode of the Dirt on Growing podcast. Hope everybody's having a great day. Today, we're going to be talking about two microbes that protect plants better than chemicals. At least that's the thumbnail for this podcast episode. What I really wanted to talk to you guys about was the consortium. Usually, I talk about Trichoderma and Bacillus subtilis. Today, we're going to be talking about the combination of Trichoderma and Beauveria bassiana. And the reason why I wanted to specifically talk about these microorganisms today was because I have been using them in my biological crop steering program for years now. And I recently went to Puerto Rico to CanMed. And, they have a bunch of different types of fields of science that are applied to cultivation. And they were talking about these specific microorganisms and how they actually function endophytically. So these particular microorganisms, both Beauveria bassiana, which Beauveria bassiana is more widely known as an insect parasitoid, right? It's more widely known as a fungus that causes white muscadine disease that parasitizes a wide variety of insects. So it's used typically for integrated pest management applications. Similarly, the Trichoderma, which is a fungus that's used for biocontrol protection and for outcompeting pathogens. What the new science has shown is that both of these microorganisms actually work their way through the root system into the vascular tissues of the plants. And so what's happening is both Beauveria bassiana and Trichoderma are actually colonizing the xylem of the plant, right? And so I've seen these new research studies that are coming out that are confirming a lot of the things that I've been saying for years about biocrop steering, which is great on my part because I kind of already knew intuitively, but now we're getting to see data. Now, when we think about what these different types of microbes do in soil, right? From a biochemical standpoint, from a metabolic standpoint, the types of metabolites that these things produce, and we apply those same scenarios internally inside of the plant, right? So these things are actually growing and living inside of the xylem. And so when the plant is uptaking water and nutrients, these microbes are acquiring those nutrients and that water, and they're multiplying and they're procreating inside of the actual plant, and they're moving throughout the vascular tissues of the plant. And look at, you can't avoid the production of metabolites, right? Secondary metabolites are produced as byproducts of microbial metabolism, which means that those microbial metabolites are inside of the plant and they're getting transported to different parts of the plant. So phytohormones, right? So I always wondered about, okay, well, what happens to a phytohormone in soil? Does it actually get uptaken by the root system? Is it broken down by the microorganism? And now what we can theorize, right, is that these phytohormones are actually being produced internally by the microorganisms themselves and being transported to different parts of the plant to induce certain responses, whether it's acquired resistances, whether it's signaling, whatever it might be. These microbes potentially could be what is responsible for all of this happening. And so when we're talking about also working endophytically and having these microorganisms transported through different vascular tissues into the plant. Well, what happens to, let's say, an insect that's feeding on that plant, right? If these microbes are working their way into the vascular tissues and you have some type of root aphid, or like an aphid, right? Or something that is sucking on that thing, like aphids, they'll go to the vascular tissues and they'll penetrate those because they want those water and nutrients. Now, if those same tissues are being colonized by these microorganisms like Beauveria bassiana, and this Beauveria bassiana is able to come in contact with that insect, in theory, right, that insect should become parasitized and it should stop the attack because this microorganism is working endophytically inside of the plant. The same thing with like Trichoderma. If this thing is living inside of the plant, and then you have some attack from something like powdery mildew, for instance, which is trying to penetrate into cellular compartments so it can suck out fluid. If we have these microorganisms that have colonized all these different parts of the plant, will it be able to elicit a response to combat that? Can it tell the plant internally, hey, this is an invading pathogen, send me out there along with your other defense chemicals to stop this thing from happening? All these things are all then become in the realm of possibility when we're able to see that these microorganisms are colonizing and procreating and living and producing metabolites internally inside the plant. It's absolutely wild. I think what we're going to end up seeing is we're going to end up seeing more science that comes out on the actual processes that are happening, the different types of chemical cascades that are happening when these are producing enzymes or proteins. What types of metabolic cascades are they triggering internally inside the plant? We're going to be able to see that through fields like ionomics, through looking at leaf tissue and sap. One of the cool things is that I don't talk about a lot, but there's a lot of cool stuff that's happening in the background. And we have a project. I worked with Medicinal Genomics to identify this spot pathogen, which I initially thought was leaf septoria. It turned out to be alternaria. And I also sent that same sample to another lab here in Oklahoma, over by OSU in Stillwater. And there is a Professor Punji, who's a professor of biology at OSU, and he studies ionomics. And so we're all collaborating on a project because Cove Environmental, they're all friends of mine that work at that lab, and they got grant money to build out an entirely brand new state-of-the-art soil testing facility. So they can do pathogen testing and screening, they can do DNA testing of soil, they can do lab analysis on soil, heavy metal analysis, leaf tissue analysis, sap analysis, they can do everything. And so what we're working on collectively is looking at inoculated plants versus uninoculated plants, and looking at the ionomics, looking at the actual atoms, the atoms that are in these plants, so we can make correlations to what is happening with the induction and with these. When we're looking at the ionomics of an organism, we can see these organisms inside of the other organisms, and we can theorize and trace and track what potentially is going to be happening from a plant metabolic standpoint and from the metabolic standpoint of the microorganisms. Man, it's so fascinating. But so what I'm hoping to do is I'm hoping that I can get these research papers that I think they were published by Medicinal Genomics, and I can get them into the Dropbox. If you've recently followed me and commented on that very first post that I did, it should auto DM you a link to the Dropbox. Now, unfortunately, haters got my Dropbox as well. So I let people get access, and unfortunately, it was my own mistake. I gave them editing access. Somebody in there went in there, they deleted all of the files out of the Dropbox. So now I have to repopulate it with all of the files. Luckily, I back up a physical copy of all of the information because I read through it and highlight stuff. And so I can just take those. They're sitting on my shelf right over here. I'm going to get those and put those into the Dropbox as well. I'm going to re-look them up on the internet. So remember, if you go to my new IG page, which is fullcrop.ceo, which I think if I edit my name and tagline here, I can do that right here. Let me put this in. At fullcrop.ceo on Instagram. If you comment on that very first post, it'll auto DM you a link to the Dropbox. So anyway, microorganisms, man, these are the two. And it's really interesting because I never really thought that having two funguses together could actually increase each other's efficiency. But I started reading some new science research, or maybe it's not new, but it's new to me, where they were actually combining Beauveria bassiana with Trichoderma, and they were seeing a greater efficiency from an insect parasitoid standpoint with the combination of them together, which is really interesting because the Trichoderma doesn't have an effect on insects, right? It can outcompete pathogens, cycle nutrients, it produces hydroxamate siderophores and phytohormones, but it's never been used to control insects. Now, Beauveria bassiana is known to do that, but when you use them together, you get better efficiency, which was really interesting reading that in these science papers. And so if we can colonize endophytically our plants, xylem and vascular tissues with these two microorganisms, Beauveria bassiana and Trichoderma harzianum, we can basically protect those plants better than probably any chemical combination. So I'm going to open this up to Q&A. I hope you guys are enjoying this. You guys are probably wondering why I'm looking so dapper today. It was because we're going to be doing a little bit of filming, going to be doing some more content creation. I've taken it upon myself to learn about video editing, content creation, and all of that so I can create pillars of content that is consistent, concise, delivers exactly what I'm trying to tell people, and builds trust in my brand. And so you can see that on the new Insta page, just trying to make everything function cohesively together. But if you got questions, just drop them in the chat and we'll get to them. And also, thanks to everybody for showing up today. I know it's the middle of the week at 11 a.m. Most of us have regular jobs. Me, I don't have a regular job. My job is very unique. I was able to use cannabis to get myself to a point where I started my own business. I'm my own boss. I have multiple different companies. And while I'm not rich, because all my money is always tied up in projects, eventually it will all pay off in the long run. You would love a video over some inputs you like for flowering phase. Absolutely. That is a good topic of conversation. What's going on? All that's green. You have done well with Recharge Mammoth P combination. Man, those, I've never personally used Recharge. I've used Mammoth P. I stopped using Mammoth P because of the price point. And when I used it was years ago before I started manufacturing my own microbial consortium. Look at this bottle is exponentially less expensive than Mammoth P. Plus, it contains a wider diversity of different types of microbes: lactic acid, bacillus, purple non-sulfur bacteria, photosynthetic bacteria, and fermentive yeast. And it has a higher colony forming unit of all of those individual microorganisms than any of the single microbes in their product. So when it comes to cost efficiency, CFU counts, I highly recommend my product over. Obviously, I am biased, but you should try out my product if you like. You can replace both of those for that product, the Recharge and the Mammoth P. Chris says, dude, I'm loving the wisdom you've been dropping. I was able to get the last 5 packs. Oh yeah. Nice. Yeah, dude. The 5 for 100 deal, that's gone. I just popped some of his genetics, some Starfighter crossed to Gorilla Glue. Okay, here we got a question from Holy Toledo. Does monosalicylic acid and fulvic help speed up the process and work hand in hand? Help speed up what process is the question. So they both work to mitigate abiotic and biotic stress factors. Silica can help balance internal charge of the plant, and silica also works to strengthen cells and the rigidity of all of the cross links between carbon and boron when they're actually, making the structure of the plant, when the plant is building its structural component. In an attempt to show the speed of septoria, I sprayed a consortium of microbes well into flower, and this is the first time in roughly 2 decades that I have not seen a speck of bud rot. Absolutely, man. When it comes to treating with microorganisms to prevent things like septoria, alternaria, and botrytis, using a consortium is most ideal because you'll get dual efficiency. And also using the right types of microbes is very important. Timing and application and quantity all make a huge difference. You need to be reapplying throughout the season. Pathogen pressure doesn't stop after one application, right? The pressure is always there throughout the season, so you need to be proactively reapplying your protection. Yep, I'm going to do some filming in a little bit. I'm going to get on the green screen. Oh, wait, no. Paul's not in today. I'm going to have to do the editing instead of Paul. But yeah, here's a question from Clownfish. Would doing a soil drench with some molasses every 2 weeks be okay for feeding the soil microbes? I don't recommend feeding directly with molasses. It's better if you really want to feed the soil like biology, or really pump biology into that soil. It's better to do a compost tea or a microbial inoculant. Let me give you an example. So this product here, the Microbe Plus, right? It is made through fermentation, and our feedstock is molasses. So we use molasses. The microbes feed off that molasses, and they produce the organic acids and the enzymes and the phytohormones, and they grow and proliferate, and then they sporulate. Okay? It's better to do something like that, or do a compost tea, like where you're using basically the molasses and maybe some other inputs to basically create a microbial soup, right? And then you can inoculate that into your soil. The reason why I don't recommend using molasses directly into soil, because if your soil isn't balanced, or if you have things like fusarium, right? Even though it may not be showing, or maybe you have some type of pathogen that's not showing, and you feed it a complex carbohydrate, its populations can explode, and then it can start to express itself as physical damage, or it'll just grow to a point where the plant's defenses, can't keep up. So I don't recommend using, regular molasses. It also could, drop your soil pH really rapidly. Even if your soil buffers it, it might be too much all at once. Woody Stem says, of course there's stuff to do, but this is well spent time. DMG says, dang, brother, looking fly. Yeah, it's good to look good. It makes you feel good. It makes other people, look at you differently as well. If you're ever feeling down, and you want like just a little boost, you can always just put on your nice clothes, go out. People look at you different. It's interesting. It's psychological. But I actually dressed up because I'm going to be doing some filming. We're going to try to do 2 Brandons where I'm basically talking to myself in the same frame. Like, this Brandon's all like looking raggedy, and then I give him, you got to do this, and then bam, he's all looking spiffy, doing something. I don't know yet, but we're doing something. So I got dressed up for the moment. Let's see. Sean Riddle says caterpillars and armyworms have been a battle for the last couple of years. Man, they always are. Outdoor is always a battle. You're always battling Mother Nature. When it comes to caterpillars, though, Bacillus thuringiensis. Bacillus thuringiensis, BT. BTK, Bacillus thuringiensis kerstaki, to be precise, is what you want. And that, as long as here's the way to really go about combating those caterpillars. You need to time when you first see them. So if you need to be vigilant about timing and what you're seeing throughout the season. So if something comes in April, right? The first wave comes in April. You need to be proactive. You need to be applying microbes at that particular point, right? To decrease their populations. If you miss it then you might have a higher population later on. Right. Okay. Then again, they come out in, let's say, September. They really start hitting again really hard in September. So you need to make sure that in August you start applying, because in September they're going to start coming hard. So timing and application is really important when you're doing preventative. How long prior to planting should the first bacteria fungus mix be applied to outdoor holes? Immediately, man. I mean, you can inoculate as early as seeds, and you should if you can, right? You start inoculating seeds, the sooner that you can make associations with microbes in the root zone, the healthier and more protected your plants are going to be. So you see this across a lot of different crops, where a lot of different seeds, they'll be coated. They'll either be coated with some type of biostimulant, or they'll be coated with microbes, and it's to help, get better germination rates, healthier starts, have better resistance to stress. Same thing. You can start treating early plants right away. And I'll give you an example. I just popped some new seeds a couple of days ago. Once they actually were about this big, I put them into some soil. You could see them like popping up. I went in there with my sprayer, and I sprayed those solo cups that those seeds are in. Right away, man. I inoculate right out the gate. Get them going right away, man. Let's see. Did I see where Klotmis Coot brought you up on the Spartan Grown from Grandmaster LED podcast on YouTube? Spartan said he uses your fulvic and humic acid. Yeah, I think all of the people that are on the Cheap Home Grow podcast, which if you guys don't know about the Cheap Home Grow podcast, for the last 6 years, every Sunday at 6 p.m. Central Time, we go live for 2 hours with a group of other growers, and we talk about, efficiency, cheap home grow, and it's a community show. It's nothing crazy. We don't even advertise on it or nothing like that. We just get in there on Sundays. I'm not always there, but I try to show up at least every few weeks. Yeah, I got myself a haircut too, man. Gotta be looking sharp. I'm ready to go to Chicago. You know what I mean? Go get some Italian food, nice glass of wine, maybe a cognac, and a cigar. That's good living right there, boys. Let's see. Sean Riddle says, I never had issues with dampering off until last year. Now, every time I pop new seeds, I lose half of them to that. I'm thinking soil is crap. I usually get ocean forest to start them off. What's a really good mix is, which I just started some seeds, because my soil that I make is way too much for seed starts. But I just went and bought some like Roots Organic. I think it's like Lush or whatever. It's really light soil, cocoa perlite mix. But also inoculate. Try inoculating early. Try inoculating early so you don't get those things dampening off. But also, it might not even be you, man. Here's the thing. There's a lot of different people that are making seeds and not necessarily doing their due diligence when it comes to like testing. You can send your material to like Medicinal Genomics, to me. There's a wide variety of places that you can get that material tested. And I test my stuff about every 6 months. I'm actually due to send all of my library off for testing again. But fusarium it can be transported and you can get it in seeds as well. So a lot of people could be making seeds, and they could be making seeds that have fusarium or pythium or other pathogens, and they're not even aware of it. You can even pass hop latent viroid in the seeds. So there's all types of issues that can arise, not from any negligence or error on your part, but could actually come from the source of where that seed was created. So All Stars is saying, I'm thinking about running 2 inline fans into my grow tent, one outside and one inside. I tried to research it. What do you think? So I have a couple of 2x2 tents. I have actually 3 2x2 tents, and I usually just run seed runs in them, just to like look at stuff that I've made. So I can, oh, yep, this is good. It didn't harm. Oh, terps are good. I want to release this or whatever. But I have one inline fan pushing cooler air from the outside of the tent into the tent, and it's on a controller, like a VivoSun controller. So it's got set parameters. It only turns on if it gets too hot, if it's too humid, or if the VPD is incorrect, and it'll adjust those things by turning on. It can go all the way from 1 to 10 on the speed setting. And what I do is I just have one way where it's putting air into my tent, and then I have the bottom flaps on my tent like slightly open so air can pass through, so air can escape easy. Bearded Roots, you live in Chicago. I get up to Chicago every couple of months. I usually drive up there because that's where the NASA Agricultural Technologies office is. That's where Dr. George lives, and I got to go see him on occasion. I've heard that, but didn't think the breeders. Yeah, you can always ask them. You know what I mean? I haven't tested in probably like 6 or 7 months. Probably 7 months, 8 months even. I did it at the beginning of the year, so I'm ready to do mine again. But I have the same cuts and nothing's moved. I've had the same, cloning off the same cuts and stuff. So I'm in a VivoSun 2x2 tent with the 100-watt light it comes with, and I'm still able to grow some fire for my first grow if I have fire genetics. Yeah, absolutely. I plan on doing living soil in a 7-gallon pot with your products. Dude, okay, so I am Louisiana. I want you to go and check out the YouTube videos that I've been updating almost like every other day on that exact VivoSun 2x2 tent setup. And what I'm doing is I'm running 9 plants in square 1-gallon pots. So I have the whole tent filled up, and I just flipped plants when they were about this big. So I flipped them when they were clones, and I put no-veg time on them. And I'm at day, I think, 18 now. You should definitely check that out. You'll get a lot of information from it. You can get genetics on my Seed Vault website, Bokashi Seed Vault website. Shipping is outrageous. I think that we have pretty good rates on our shipping. It's like calculated from like the post office. So raw genetics. I've got a website too, you guys. Look at www.bokasheedvault.com. I've also checked, dude, I've got so much cool stuff. Let me see. Let me see if I can take this camera. Yeah. Over here, real quick. You can see right over there. These are all unreleased as of now. But I have several new chem varieties, and I have several new Starfighter cross to Mac varieties that I'm testing right now. But anyway, we've been here for half an hour. I'm going to let you guys get back to work. I got to get back to work. Thanks for hopping on here. Make sure you guys go check out the website, and you can always DM me on Instagram or email me if you guys have any questions. Until next time, I'll see you guys later.